Today I'm going to be talking about smooth endoplasmic reticulum. So what is smooth endoplasmic reticulum? Smooth ER is an organelle found in both animal and plant cells. When looking at the definition of endoplasmic reticulum, endoplasmic means in the cytoplasm, and reticulum means a network. So the definition of endoplasmic reticulum is a network of tubules within the cytoplasm of a cell. Smooth endoplasmic reticulum was discovered by Albert Claus, Keith R. Porter, Brodie Meskers, and Ernest F. Fulham. The structure was discovered using the newly developed electron microscope. The structure of smooth ER changes from cell to cell. The cell's function is what determines the size and the structure of the ER membranes. Some cells don't have any ER. For example, prokaryotes don't have any ER. Other cells that synthesize and release a lot of proteins need a large amount of ER. For example, pancreatic cells and liver cells need a high amount of ER. ER membranes are folded and stacked on top of each other and connected to the nucleus. The interior of smooth endoplasmic reticulum is called lumen, which is surrounded by a phospholipid membrane. The smooth ER consists of tubules that are interconnected and flattened stacked patches called cisternae. Much like the structure, the function of smooth endoplasmic reticulum also differs cell to cell. Smooth ER synthesizes and distributes different substances based on the type of cell. For example, in muscle cells, calcium is needed for muscle contraction, and due to this, the ER membranes are used to produce and release calcium ions. The smooth ER performs this function by pumping calcium ions from the cytosol into the ER lumen. Endocrine cells, such as the pancreas, regulate hormones. The smooth ER and endocrine cells produce steroids, estrogen hormones, and testosterone hormones. The smooth ER can also be used to produce lipids and fatty acids and metabolize carbohydrates such as sugars and starches. Smooth endoplasmic reticulum cannot take a role in producing proteins due to the lack of ribosomes that are embedded in other ER membranes such as rough endoplasmic reticulum. Another function of smooth endoplasmic reticulum is detoxifying harmful substances and changing it into helpful substances. This is usually done by converting harmful substances to safer water-soluble products. It usually involves adding hydroxyl groups to drug molecules. Smooth endoplasmic reticulum makes fats that are used for the production of phospholipids. In liver cells, smooth ER enables glycogen that is stored in granules as granules in the external surface of smooth ER to be broken down to glucose. As for the function with other parts of the cell, the lipids and hormones made by the smooth ER are sent to the Golgi and packed there to send to other parts of the cell. ER membranes are located throughout the cell. They are folded and stacked on top of each other and connected to the nucleus. The main difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum is the rough endoplasmic reticulum has a rough exterior covered in ribosomes that are used for protein synthesis. The smooth ER does not have a ribosome coating and appears as a series of interconnected smooth tubes. Another difference between the smooth ER and the rough ER is the location in the cell. Although endoplasmic reticulum is located throughout the cell, the rough ER is located closer to the cell, whereas the smooth endoplasmic reticulum is located further. In addition, the smooth ER is tubular and the rough ER is flatter. A difference in the function of rough endoplasmic reticulum and smooth endoplasmic reticulum is the interaction with protein. Smooth ER is not used in the synthesis of protein like rough ER is. However, it does play a big role in the transportation of proteins to other parts of the cell. The reason smooth ER cannot be used in the synthesis of protein is because it lacks the ribosome coating that rough ER contains. Thank you.